Hello guys, and welcome to our video on the cell cycle. So what is the cell cycle? The cell cycle is essentially the sequence of events that lead up to and include mitosis. So as you can see here in figure 6.1, we have a diagram of the cell cycle. So it consists mainly of the G1, the S, the G2, and the M phases. And you might have heard the term interphase when talking about mitosis. So interphase refers to G1, the S, and the G2 phase. Also, one more thing here. The G1 phase stands for gap 1 phase. Same thing with the G2 phase, gap 2 phase. The S phase stands for synthesis. This is where your main DNA synthesis, histone synthesis, those kinds of things will happen as per this diagram. Alright, so we can often get confused a lot by the vocabulary used when we talk about mitosis and meiosis later on. So let's just go over some vocabulary. So what is a chromosome? A chromosome is essentially one DNA molecule consisting of two strands, as does every DNA molecule, because as we know, DNA is a double helix. And that DNA molecule is packaged with histone proteins, which are positively charged, by the way. So we know that DNA has a negative charge because of the phosphate group, as we covered earlier. And so the histone is positively charged. And so these histones and the DNA are going to pack together very tightly because we know that opposite charges attract. So that's what forms a chromosome, DNA and proteins, which are histones. Then let's go on to our chromatid. Our chromatid, as you can see in this diagram, it occurs after the S phase, after the replication of the chromosomes. The chromatid is sort of like this piece, this half of a chromosome. So when a chromatid separates, we consider it as its own chromosome. But when it's attached to another chromatid, we consider both of those as one chromosome. That can be a little confusing, but you'll get used to it eventually. Then we have the centromere. So the centromere is an organelle, which is basically, it's not an organelle. It is simply the attachment point of the chromatid. So as you can see, this little bend, sort of like a waist-like bend in these chromosomes, that is the centromere. Then we have chromatin. So chromatin is simply the material that forms a chromosome. So DNA and histone proteins combined, we like to call that chromatin to refer to the material itself. Then we have mitosis, which is the division of the cytoplasm. It results in two different daughter cells from one daughter cell, and the DNA is replicated as it is. There's no like genetic variation or anything like that. Meiosis we'll cover later, but that involves gamete formation, and it adds genetic variation. So that's just to clear up some doubts that we may have had about the vocabulary. Alright, so let's start with the first major phase of the cell cycle. That is the G1 phase. The G1, like I said, phase, it stands for gap 1 phase. So, again, the G1 phase is the first part of interphase, and the average cell is in this phase. This is when they grow, they execute their function. So, for example, a muscle cell, or a skin cell, for example, in G1 phase, is going to, be guarding the interior of protecting the interior of the body from, like infection, or preventing other substances from entering the body that shouldn't. It's essentially the purpose of the skin, right? So that's what a good example of a cell that's in G1 phase is going to be executing its functions. Then the main checkpoint of the cell cycle occurs after G1 phase has occurred. This is really important because most cells that will advance through the G1 checkpoint will end up advancing through the entire cell cycle. So a checkpoint is essentially a set of criteria that a cell has to meet in order for it to continue onwards with cell division. And it's really important because if the checkpoint fails, if it allows for a cell to continue dividing when it's not ready to divide, whether it's because the cytoskeleton isn't set up correctly, or the DNA has not been replicated, or something like that, if the checkpoint allows that to happen, that can develop into cancer sometimes. So these checkpoints are really important. Okay, so some cells are in G0 phase, and usually this is because they failed this checkpoint. So it may have evolved evolutionarily to prevent cells that may have become cancer cells or something like that from causing too many problems. But in addition, there are some regular cells like neurons or muscle cells that don't divide very often. So those cells are usually in G0 phase. They don't really participate in much of the cell cycle. Alright, so next we have S phase. The S phase is just really simple. It's just where DNA replication occurs, and also histone replication occurs and things like that. But the primary thing you'll need to know about S phase is that DNA replication occurs. So here we go from 46 chromosomes or chromatids to 92 chromatids because we have the DNA replicating, right? So then we have G2 phase. So there really isn't much to know about G2 phase other than the fact that this is sort of like the final checkpoint before mitosis kicks off. So the cell checks through the DNA for errors because the DNA has just been replicated. Although errors in replication are relatively rare, when they do happen, they can have devastating effects. So most organisms have evolved mechanisms to address that, to be able to fix these problems. And in addition, MPF, which stands for maturation promoting factor, but it's sort of a molecule that accelerates or begins mitosis, initiates mitosis. And so it's officially called maturation promoting factor, but it may be nice to think of it as mitosis promoting factor because that is essentially what it is, and thinking of it that way also helps you remember what MPF is. So MPF is mainly formed from a cyclin-dependent kinase, which is a certain protein, and then we have cyclin. So the cyclin, it begins to peak. It begins being synthesized in G2 phase, and then it peaks at anaphase, and then after that it sort of gets degraded. So cyclin-dependent kinase, however, it stays constant. It doesn't really get degraded or made that often, but the cyclin is what gets addressed. And so if the cyclin gets broken, then MPF sort of doesn't exist anymore. It's just a cyclin-dependent kinase, and that means that we won't have any MPF or as much MPF. Finally, we get to mitosis. So mitosis is made of 4 major phases. There's prophase, there's metaphase, there's anaphase, and there's telophase. So in prophase, what happens is that the chromatin, which again, if you remember, is the DNA and the histone proteins, the material that makes up these chromosomes, it begins to condense into a denser form we like to call heterochromatin, euchromatin. And so when it begins to condense, the nuclear envelope also disintegrates. And this also forms these chromosomes which you can see under the microscope. So normally chromosomes are not the way you see them under the microscope. In fact, most of the chromosomes that you have seen, they actually are taken in metaphase or during cell division, those pictures. So usually chromatin is just really loosened out, hard to see under a microscope, but in prophase they begin to condense, nuclear envelope disintegrates. So in metaphase, by metaphase, the spindle apparatus is formed. It begins forming in prophase, but it finishes forming up in metaphase, and the spindle apparatus sort of provides structure for the cell during cell division because, the cytoskeleton is really disrupted. It's beginning to disintegrate, so there needs to be something to provide structure to sort of conduct the whole process. That's what the spindle apparatus is here for. The centrosome, which is an organelle of the cell, is the main organizing area. We'd like to call it a microtubule organizing center during this process. So the apparatus aligns the chromosomes as what is known as a metaphase plate, which is essentially the middle of the cell. And then we have anaphase. So in anaphase, the sister chromatids, which are the 2 arms, as you can see here, of the chromosome, they split into what become two different cells. And here also the MPF levels begin to fall drastically because the cyclin, like I said, gets destroyed. And then in telophase, the cells finally pull apart and become their own cells, their own functional units. And then the nuclear membrane reforms. The chromosomes sort of like loosen out, they become normal again, and then you have the cytoskeleton also forming in the cells. These two independent entities now go about the process one more time. So again, like I said, MPF levels are going to increase from G2 phase all the way up to anaphase, and then anaphase is going to be at its highest levels, and then it will dramatically fall. So in this video, we covered the cell cycle, starting with some basic vocabulary, and then we went into the G1, S, G2 phases, and then finally we addressed mitosis. So I hope you guys enjoyed our video on the cell cycle, and I hope you watch next time.